Good morning. My name is Vikram Gadagkar, and I'm an assistant professor at the Zuckerman Mind Brain Behavior Institute at Columbia University. It's a great honor to be asked to speak at Lab Roots 2024. Since I'm coming to you from New York, I thought I would begin with a New York story. The great pianist Arthur Rubinstein was walking along the streets of New York one day when a pedestrian approached him and asked him, "How do I get to Carnegie Hall?" Rubinstein pondered this for a second and said, "Practice." Indeed, many of our behaviors are not innately programmed but are acquired through a process of trial and error or practice. We all know that learning to play tennis or the piano can take years and sometimes a lifetime of practice. Much of our understanding of the neural mechanisms underlying trial and error learning comes from the study of animals engaged in various tasks motivated by primary rewards like food or juice. Put a rat in a box with a lever, and he will learn by trial and error to press the lever for a food reward. Classic experiments in the 1950s by Olds and Milner showed that we can remove food from the equation if we hook up the lever to an electrode with which the rat can self stimulate his dopamine neurons. If we do this, the rat will learn to press the lever not for food but for a squirt of dopamine. We are all familiar with the addictive properties of dopamine, but dopaminergic signals are also ideally suited for trial and error learning. These data show the response of a midbrain dopaminergic neuron that projects to the basal ganglia of a monkey that has been trained to associate a light cue with a juice reward 1 second later. Time is on the x-axis, each dot is an action potential, each row is a trial, and what is shown on top is the firing rate histogram. Since the light is present at random, the appearance of the light is a better than expected outcome, or what we call a positive prediction error, and is signaled by a phasic activation in firing rate. Now, 1 second later, when the monkey is expecting juice, if the reward is withheld, that would constitute a worse than expected outcome, or what we call a negative prediction error, and is signaled by a phasic suppression in firing rate. In other words, this neuron can be thought to encode what is called reward prediction error, or the difference between the actual reward and the predicted reward. These phasic activations and suppressions are thought to positively and negatively reinforce preceding motor acts, thus leading to learning. In other words, whenever dopamine goes up, that's telling the brain, whatever you did just now was great, do that again. And when dopamine goes down, that's telling the brain, whatever just happened was not too good, don't do that again. The principle of dopaminergic reward prediction error has been highly influential in neuroscience. However, most of our motor skills, like speaking or playing a musical instrument, are not learned in the pursuit of immediate food or juice rewards, but are instead learned by comparing ongoing performance to internal goals. Imagine, for example, that you're trying to learn an 8 time-step song on the piano. And you want to play C sharp at time step 3 and F sharp at time step 6. Now, while you're practicing this piece, if you happen to play C sharp at time step 6, your brain immediately recognizes this as an error or a mistake. The point here is that there's nothing intrinsically good or bad about the sound of C sharp. It is simply that you wanted to hear it at time step 3, but did not want to hear it at time step 6. So the question I was interested in is, how does the brain evaluate such internally guided motor sequences? For this, we turn to the zebrafinch. First of all, zebrafinch songbirds don't learn to sing for food or juice rewards. The adult zebrafinch song is a naturally learned, highly stereotyped motor sequence. Here is a spectrogram of the zebrafinch song. Time is on the x-axis, frequency on the y-axis, and the color represents spectral power. You can see that the song consists of distinct syllables A, B, C, and so on that he repeats again and again and again. A baby finch is not born being able to sing like this. All he can do, just like a human infant, is babble. The father finch sings his song to the baby finch. The baby remembers the father's song and then spends the next 2 months as he's growing up learning to make a copy of the father's song through a process that looks very similar to trial and error learning or practice. How does the zebrafinch brain manage to accomplish this remarkable feat? Now, we know that behavior is driven by neural circuits, and much of our understanding of the neural circuits underlying reward-based trial and error learning in mammals comes from the study of basal ganglia thalamocortical loops including dopaminergic projections to the basal ganglia. It turns out that these neural circuits are evolutionarily conserved, and so songbirds also have these basal ganglia thalamocortical loops, including dopaminergic projections to the basal ganglia. We just sometimes call them by different names. One of the great advantages of the songbird system is that the songbird brain contains a discrete set of interconnected brain regions called the song system that's dedicated to singing, including a dopaminergic projection from the nucleus VTA to area X, which is the singing-related basal ganglia. Inspired by the mammalian literature on external reward prediction errors, we and others hypothesized that dopamine neurons in VTA might convey a conceptually similar error signal for song learning, even though song is internally evaluated. To test this hypothesis, I had to record from neurons in VTA, but not just any neurons in VTA, but specifically those neurons in VTA that project to area X. To do this, I used what is called antidromic identification, where I put a stimulating electrode in area X and a recording electrode in VTA. I stimulated area X to look for a backpropagating signal along these axons to be picked up on my recording electrode. If I saw these signals, then I knew that the neuron I was recording from projected to area X. I had to record these neurons while the birds were singing and while I fooled the birds into thinking they made a mistake. To do this, I recorded these neurons during a paradigm in which 50% of the renditions of a particular target syllable were distorted with auditory feedback. To ensure that the feedback sound had similar spectral and temporal properties as normal zebrafinch song, I took a 50 millisecond snippet of a different syllable and played it over the target syllable. What do I mean by this? So let's say the bird song has 4 syllables, A, B, C, D, which sounds like chikuwa, and I take the first part of syllable A, which sounds like ch, and play it over syllable D. So the bird is singing along, chikuwa, 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 but what he hears is chikuwa, 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 depending on whether or not syllable D was distorted with feedback. The idea is that a distorted syllable sounds worse than expected to the bird and should cause negative prediction error signals. It turned out that recording these neurons was a big challenge, and it took us several months before we could start recording these neurons. Here is the first neuron that we were able to record during this paradigm. Let's look at the distorted trials first. So here we have the distorted spectrogram. You can see the different syllables A, B, C, D, and so on, and you can see that syllable D has been distorted with feedback. Below that is the example neural trace. Each line is an action potential, and the yellow box represents the time during which the feedback was presented. Below that, we have the raster plot and the firing rate histogram. Again, time is on the x-axis, each line is an action potential, each row is a trial. Looking at these plots, you can see that this neuron shows a distinct suppression in firing on every single trial immediately after feedback. This is exactly the kind of signal we were looking for, and this is consistent with the syllable sounding wrong. Now, what about the undistorted trials? Once again, we have the spectrogram, but notice how syllable D here has been left completely untouched. Below that, we have the example neural trace, the corresponding raster plot, and the firing rate histogram. Looking at these plots, you can tell that this neuron shows a distinct activation in firing at precisely the time point in the song when a distortion might have occurred but did not. This is consistent with the syllable sounding right. Now here are the same data plotted a little differently. So once again, we have the undistorted and distorted spectrograms, example neural traces, raster plots, and firing rate histograms. If you focus your attention on the rate histograms, you can tell that this neuron has a nearly identical firing rate at all time points in the song, except immediately after target time, when there is a distinct suppression after distorted syllables and a significant activation after undistorted syllables. Now, the suppressions are easy to understand. We distort the syllable, it sounds wrong, it sounds worse than expected, and so there's a suppression in dopamine. But why is there an activation on undistorted syllables when we do nothing at all to these syllables? Our interpretation is that because we were distorting the syllable D, for example, half the time, the expected quality of syllable D was lowered for the bird. So when the bird heard an undistorted syllable D, even though it was completely untouched, it actually sounded better than expected. And that is what is signaled by this phasic activation. So once again, we have a better than expected and worse than expected dopamine signal, but not for primary rewards like food or juice, but for performance outcomes. That is, whether your own performance was better or worse than you expected it to be. So we call these kinds of error signals performance prediction error signals. Now, we were very excited about these results because first, it identified the long sought after error signal for song learning. And more broadly, it showed that the principle of dopaminergic prediction error can generalize to the learning of complex motor sequences that are not learned for primary rewards, but are instead learned by comparing ongoing performance to internal goals. However, one of the questions that came up was, how do we know that these neurons actually care about the quality of song and are not simply responding to the presence or absence of this feedback sound as if it were an aversive stimulus? We presented several lines of evidence to argue that these neurons actually care about the quality of song, but this question motivated me to ask if we can test directly if these neurons care about song quality. So what do I mean by this? So let's say this is the raster plot that you have already seen. Now we know that there are very few spikes here because the quality of song that preceded this time point was low because it was a distorted syllable. Similarly, there are lots of spikes following undistorted syllables because the quality of song preceding that time point was high. It was undistorted. Now, what if we look away from the regions that we distorted? For example, there seem to be a lot of spikes on these trials. Is that because the quality of syllable A happened to be high on those trials? Similarly, there are very few spikes on these trials. Is that because syllable A happened to be sung worse on those trials? In other words, can the song quality at a particular time in song predict the number of spikes after a brief latency? To answer this question, we used a Gaussian process model approach, which I will describe here in cartoon form. So let's say we have 3 song renditions and 3 simultaneously recorded spike trains. The first step is to parameterize the song into different song features like pitch, entropy, and so on. We used 8 different song features, but I will only show 2 of them here for simplicity. So the question now becomes, can the value of a song feature in a particular song window predict the number of spikes in a suitably chosen spike window? So we first construct a model of spike count as a function of pitch alone, then a model of spike count as a function of entropy alone, and then a model of spike count as a function of both entropy and pitch with a penalty for an extra parameter. In this way, we construct models of spike count as a function of every parameter and every combination of parameters. Each of these models gives us a goodness of fit or an R² value, which we use as model weights to construct a combined multidimensional model of spike count as a function of all the features. This combined model itself gives us a goodness of fit or an R² value. Now we can repeat this procedure for all possible latencies. So what does that look like? So imagine we have 60 song renditions and 60 simultaneously recorded spike trains. For a particular song window spike window pair, we can do what I just described and plot the R² value on this plot. The x-axis here is the spike bin onset and the y-axis is the song segment onset. Now, I can consider all possible spike windows and all possible song windows and generate an entire matrix of such R² values. And I can then repeat this procedure for all the natural syllables in the dataset. That is, all the syllables that were never distorted with feedback. And then we can ask, are there good fits? That is, are there relationships between spiking and song? And if so, at what latencies do they occur? So here is the summary plot of this analysis. The x-axis is the song spike latency, That is the time between the song window and the spike window. And the y-axis is the normalized count of good fits. The blue line represents the real data. The black line and the gray shading represent the mean and standard deviation of shuffled datasets, where we shuffle the identities between song renditions and spike trains. Looking at this plot, you can tell that there seemed to be a preponderance of good fits at a brief positive latency of about 50 milliseconds. A positive latency is encouraging because that suggests an evaluative process. That is, the spikes that care about how the song was sung were produced after the song was sung. But does the latency of 50 milliseconds make sense? I have taken the plot that you've already seen from my previous feedback experiments, and I have stretched the x-axis to match the x-axis below. And you can tell that the latency of about 50 milliseconds exactly matches the kinds of latencies we observed in our feedback experiments. That is, even in our feedback experiments, these dopamine neurons started caring about whether or not there was feedback about 50 milliseconds after feedback onset. Now, while I was recording these neurons, I also recorded a bunch of neurons that did not project to area X and correspondingly did not have an error signal. So we reasoned that if we do a similar natural song analysis on these neurons, we should not see this kind of a peak. And in fact, when we repeated this analysis on non-area X projecting, non-error responsive neurons in VTA, we do not see a similar peak at 50 milliseconds. These results suggest that these dopamine neurons, in fact, seem to be evaluating natural song independent of any external feedback or perturbation, thus validating our feedback experiments. Now, everything I've told you so far is about practice. But the entire reason we often practice a motor skill is so that we can perform in front of an audience. And we know that practice is associated with exploration and variability that we harness for learning, whereas performance is characterized by exploitation and stereotypy of learned behavior. I was telling some of my musician friends about these results, and they claimed that they treat mistakes differently during practice and performance. During practice, they pay a lot of attention to their mistakes because they want to learn from them. But if they have perfected the song and they're performing in front of an audience and then they happen to make a similar mistake, they might brush it under the carpet because, the show must go on. This led us to wonder, can the brain actually handle motor errors differently during practice and performance? And it turns out that zebra finches are once again a great system to ask this question. Because male zebra finches practice their songs alone with the ultimate goal of performing to a female. It is, after all, a courtship song. And in fact, singing alone produces a slightly more variable song compared to singing to a female, or what we call female-directed song, which is more stereotyped, consistent with this practice versus performance idea. So to test what happens to this error signal, I repeated my previous set of experiments, but now, first when the males were singing alone, and then I showed them a female, got them to sing to the female while I'm still recording the same dopamine neurons to see what happens to the error signal. So here is a neuron that we recorded during this paradigm. Let's see what happens first when the male is singing alone. So when the male sings alone, just like before, we see an error signal, a significant suppression after distorted syllables and a significant activation after undistorted syllables. But then, when the male starts singing to the female, you can see that this error signal that was present during practice is now reduced and often completely turned off when the male sings to the female. This suggests that these dopaminergic error signals can indeed be gated off during performance, and is the first demonstration of the social modulation of dopaminergic error signals. So this led us to wonder that perhaps what is happening is that your self-evaluation system, which is active when you're practicing, has been turned down when you're performing. So maybe you don't care so much about your own mistakes when you're performing to an audience, but perhaps you care a lot about how the audience is responding to you. In this case, the audience is the female. And these female zebra finches don't sing, but they make these short vocalizations called calls, which sound like eh, 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 eh. So then we wondered, maybe these dopamine neurons care about the female calls. To do these kinds of experiments and a lot of other experiments that came up, it was impossible to do this with the electrophysiology that we were using until now. Because with this electrophysiology, you could hold these neurons only for a few minutes, but social interactions can persist for much longer. To solve this issue, we developed dopamine photometry for the first time in singing birds. In dopamine photometry, what we do is we use a virus to inject a modified dopamine receptor into area X. And when dopamine binds to this modified dopamine receptor, it releases light or fluoresces. And this light can be picked up with an optical fiber that you can implant into area X. So now the amount of light that is coming out of area X becomes a proxy for the amount of dopamine that is being released into area X. There are two main advantages to dopamine photometry. First is that dopamine photometry allows you to directly measure the dopamine released into area X instead of measuring the action potentials of VTAX neurons. And second, dopamine photometry can monitor dopamine for over much longer timescales, from weeks to months, whereas electrophysiology could only do it for a few minutes at a time. So the first question we wanted to ask was, can we recapitulate the error signal in dopamine release? And the answer to that is yes. So I have plotted the data here in a very similar way. The only difference is that instead of action potentials, we now have fluorescence signals. But you can see, looking at the histograms below, that once again, there's a significant suppression shown in red after distorted syllables and a significant activation shown in blue after undistorted syllables. So once again, we have the error signal in dopamine release in area X as well. Next, we wanted to ask if the error signal that we find in dopamine release also gates during performance. Does it also get reduced when males sing to females? And in fact, that is exactly what we found. So you can see here that there's a robust error signal on the left when the males are singing alone, which gets substantially reduced when they sing to the female. So now we can ask if these dopamine neurons become less sensitive to the male's own mistakes, but more sensitive to potential feedback that the females might be giving them. So we then recorded the response of these dopamine neurons to female calls. You can see from this plot that dopamine does respond to female calls. When the call happens outside of singing, there is a small response shown in blue, but you can see that there's a much bigger response shown in orange when the calls overlap with the male song. These results suggest that dopaminergic error signals can retune to social feedback during courtship. The big take-home message, I think, from all of these results is that satisfying internal goals in the pursuit of learning activates the same reward systems that have been so extensively studied in neuroscience. So the next time you're really thirsty and somebody unexpectedly gives you some juice, the neural circuits in your brain that are activated might be the same circuits that are activated when you're trying to learn to play the guitar and you happen to hit the right note. So the next time you're practicing your 8 time step song on the piano, and you happen to do a better than expected job at time step 3 and a worse than expected job at time step 6, there is perhaps a dopaminergic projection to your basal ganglia that does this. A phasic activation at time step 3 and a phasic suppression at time step 6. But if you now practice this piece and become an expert and decide to perform in front of an audience, then your self-evaluation system might be turned down, and if you make similar errors during performance, your dopaminergic signal might look like that. However, now your dopamine system might become much more attuned to feedback from the audience. In my own lab, the big question we're interested in is, how do brains interact? In other words, what are the neural mechanisms for social behavior and communication? As we saw in the talk, many of our socially relevant motor skills, like speaking or playing an instrument, are learned through a process of trial and error or practice. They are then performed to a socially relevant target audience, and the audience might show a preference for certain features like stereotypy or virtuosity and might provide feedback to the performer. We can ask questions at each of these levels in zebra finches because male zebra finches practice their songs alone with the ultimate goal of performing to a female, and female zebra finches need to evaluate the quality of male song and might even provide feedback to singing males. The big questions we're interested in each of these levels are first, now that we have access to this error signal, what are the causes and consequences of this performance error signal? In other words, how is this error signal put together? How is it constructed? And how is it then used to actually learn the song? How does the brain switch between practice and performance? And finally, how do we evaluate the actions of others? I'm now going to give you a brief glimpse into three different ongoing projects in the lab. The first is asking how is reinforcement learning actually implemented in biological systems during the actual learning of a behavior. Everything I've talked to you so far were experiments in adults that had already learned their songs and were maintaining their songs. Now we can begin to ask how do juvenile zebra finches actually learn their songs? So what we can do is we can record every single vocalization produced by a juvenile bird as it's learning to sing. We can use neural networks called variational autoencoders to characterize each of these vocalizations. And we can use the newly developed dopamine photometry in singing birds to monitor dopamine transients that's associated with every single vocalization. And then we can ask questions like, does dopamine actually guide vocal learning during the actual learning of a motor skill? Now, neuroscience has taught us a lot about how the brain produces behavior, but we know very little about how one brain evaluates the behavior produced by another. Songbird neuroscience has also primarily focused on singing males, and the female brain and the neural mechanisms of mate choice have been largely neglected. Female zebra finches don't sing, but they need to evaluate the quality of male song and decide whether to mate with the male or not. And since these birds are monogamous, this is one of the most important decisions in a female finch's life, yet we understand very little about how she's actually doing it. One of the features female zebra finches care about is song stereotypy. That is, how consistently can the male hit the same notes again and again and again. But evaluating song stereotypy is a complex task. She needs to remember the first few renditions of the song, compare the subsequent renditions to the first renditions, and estimate some kind of metric of variance. How does she do this? Now, it turns out that female zebra finches, even though they don't sing, have a song system in their brains. And one hypothesis is that females are using their song system and associated auditory areas in their brain not to produce song, but to evaluate the songs produced by males. We are now recording from different song system nuclei in the females to test such hypotheses. And more broadly, we're thinking of this as a model for how we evaluate the actions of others. Finally, in a third, more ambitious project, we want to understand the neural mechanisms underlying the formation and maintenance of social networks. As we know, many human social networks are formed and maintained through conversations and vocal interactions. Zebra finches also live in flocks and they communicate with each other through a variety of calls and songs. In collaboration with Richard Hanloser at ETH, we are developing a new paradigm called neuroflock, where we have an arena with a group of zebra finches and we use microphones and cameras to record the position, texture, and vocalizations of this interacting group of birds. For example, we use machine learning to do social behavior tracking. Each zebra finch wears an individual backpack to record individual vocalizations, and we're using machine learning based syllable labeling to automatically annotate every vocalization produced by this group. The goal is to then combine this with neural recording and manipulation to begin to understand how the brain allows for social networks to be formed and maintained and answer questions such as what are the neural mechanisms underlying courtship and monogamy. I'd like to thank my lab at the Zuckerman Institute at Columbia, my funding sources, and I thank you very much for your attention.